Okay, can you all hear me?
Yes.
Okay. Today we will mainly revise paper 6 and anything that you need revision. Okay, so I'm going to share my screen and start with paper 6. If you have any questions, today is the time to answer any question you want. Fine, so we will start with paper 6. And I just want to bring to your notice that some investigation questions can also be available in paper 4. Fine, now paper 6, by now you can solve a complete paper 6 exam because paper 6 has basic criteria. Some of the calculations that you have to know for basics for paper 6, number 1 is the magnification. Let's revise it together. Who will remind me, how do we calculate for the magnification equation? Can you all see my screen, please? You all can see my screen? I hope it's clear. Yes, can you tell me what's the magnification equation?
Image size over actual size.
Yes. This is the easiest way to calculate the magnification of any image or object. I is the size of the image. AE is the actual size, actual or the real size. And the magnification is how many times it is made bigger, okay? Now, in case you forgot, if you have or you are asked about the magnification, then you will put your finger on the missing variable and you should divide the image over the actual or the real size. Some of you are still confused up till now. The image size is the size of the picture in front of you. And the actual size means the real size. Now, sometimes they will play about with the units. The units, they have to be the same, okay? And you have to choose the smallest unit that is in the question. First, you will measure the image with a ruler, and you should have a ruler and a calculator on the exam. You will measure any image size. It will be in which units?
Millimeter.
Okay, if your ruler has a millimeter unit, fine, but usually it is in centimeter. How do I convert from centimeter to millimeter?
Times 10.
Yes, and then if they ask you, or there is a smaller unit than that, it will be in micrometer. How do I convert the millimeter to the micrometer?
Multiply 1000.
Yes, multiply by 1000. This looks very easy, but I still see lots of mistake. And in case you forgot, magnification has no units. Magnification needs to be a whole number. The magnification can never be a negative number, and it can never be a decimal. And it is always times 30, times 300, times 40, whatever. Okay? So there is no units for magnification. There is always the times. Now, again, in paper 6, don't tell me that magnification is in the lesson classification, so it will not show up. No, it will. Okay, another thing that is important and very common and available in all paper 6 is the drawing. You will be asked to draw, and the drawing is an easy question out of 4 marks because it has also some criteria. If they ask you to draw a picture and do not draw any cells, then do not draw any cells. Don't add a cell because you want to add something, you lose a mark. Okay, now the drawing, why is it out of 4 marks? Because 1 mark goes to the size of the drawing, it has to be big, and the lines have to be straight and thick lines, not any broken lines, no broken lines, they're not broken, no shading, don't do any shading in your drawing, and you have to get a mark for the details. In other words, the picture that you draw has to be a replica of the picture in front of you. So this is very easy. Just practice any drawing you see in paper 6, draw it, and you can send me the drawing and I'll tell you how many marks you will take for that. Okay, another factor that is very important in paper 6 is are the graphs. Graphs and graphs and graphs. Again, graphs, believe me, they are very easy and you just need to focus on the graphs. A graph, any graph in the world has 2 axes. The horizontal is the X and the vertical is the Y. First, the first step is that you need to decide what will I plot on the X and what will I plot on the Y. Any investigation question, for example, I want to test the effect of pH on the rate of a reaction. So I will be changing the pH and I will check how this change will affect the speed of the reaction. So if I will be changing the pH, what is my independent variable?
The pH.
Yes. Okay. And I don't want only one person to participate. I want everyone to rush and give me the answer because this should be very easy. We did this so many times. As long as I am changing the pH, then the pH will be my independent variable. And the independent variable is always plotted on the x-axis. It can be pH, it can be temperature, it can be concentration of solution, it can be anything. As long as I am changing one factor, that factor is on the x-axis. Okay, because if you get this wrong, everything else will be wrong. Now, my results, what I am measuring, will be the independent or dependent variable?
Dependent.
The
Dependent.
dependent variable. So it
Dependent.
could be my results. My results can be anything depending on the investigation. The rate of the reaction can be counted by the number of bubbles, how fast something or a product will be produced, etc., etc., etc. So my dependent variable on the y-axis.
Y-axis.
Excellent. Now, why do you lose marks? Because you forget the units. Please write the units. Please, please, please write the units because you lose a whole mark because you missed the units. So first, you will label your axes. You will decide where every variable will go. You will write the units. And then the first step you will do is that you will divide your graph into boxes of 5 that are of the same size. Both axes, okay? Go to your table. Let's say I have, I'm changing the pH, I will not change, and I will see how this change of pH will affect the rate of the reaction. I have to go look for the maximum value. Let's say my maximum pH is 15. So I have to make sure that 15 is close to the end of the axis, that the 15 has to be close to the end. My 15 value cannot end here, cannot even end exactly in the middle. It has to extend and end near the end of the axis. So I have to find a way to distribute my values so that my maximum value, which is 15, has to be at the end of the axis. And these values, please, they have to be equal. You have to start with a 0, 5, 10, 15. No, something is wrong. My 15 cannot end here. I want my 15 to end here. So I have to make the values a little bit smaller. That's why I have to use a pencil so that when I come to do changes, I erase. I don't mess up with my pen. 2, 4, 6, 8, 10. Let's make it longer. Okay, of course, you will have a grid that will fit your values, of course. So let's say 10, 12, 14, 16. Very good. My 15 is here. So my 15 is close to the end of the x-axis. And the same for the y. I have to distribute my values so that my maximum value is close to the end of the axis. Please, the plotting, even that gives you a grade. You plot with an X or with a dot surrounded by a circle. Okay? And to be on the safe side, join the center of the X or the dots with the ruler. But if you obviously see a curve, then join it with your pencil. If you see the dots forming a curve like this one, okay, you have an X here, X here, X here, X here, X here. You want to join it with a ruler, you will not lose marks. If you obviously see a curve, then use your pencil and join the center of the X's nicely, okay? And do not extrapolate. Do not do an extension and tell me it has to meet the 0. No, it does not have to meet the 0. It will meet the 0 only if there is a value that makes it meet the 0. Okay? Now, I want to stress on bar graphs because I don't know why you have a problem with bar graphs. With bar graphs, we do the same steps exactly. The independent variable will be plotted on the x-axis, and I do not mention or I do not forget the units. And my results will go on the y-axis also, my dependent variable, and I will not forget the units. And my bar graph, again, I do the same thing. I write a 0 here, okay, because this 0 will refer to the y-axis. I distribute my values on the y-axis equally, divide my y-axis into boxes of 5, and divide my bar graph also into boxes of 5. This will make it very easy because bars in a bar graph, they are joined or not joined. in a bar
Not
graph.
joined.
Not joined. They have to be the same width and they have to be equally distant. So to cut a long story short, I will make each bar with a width of 5 boxes and separated by another 5 boxes. So I will then meet the criteria of my bar graph and I will not make the bars fused. So I can leave the first 5 boxes if I have the space, and then make a bar here, and then leave the second, and then make a bar over there, and then leave the third, and then make a bar over here. Okay, so now my bars are definitely the same size and they are equally distant. And if the question tells you plot a bar graph, then don't use your imagination, please, and give me a line graph or a histogram. They said bar, then do bar. Okay, sometimes they try to confuse you. Usually in a bar graph, the independent variable is not a number. Usually the independent variable, let's say, is blood types. For example, blood type A, blood type B, blood type AB, and blood type O. And yes, this reminds me that the blood types, this is an example of which type of dominance?
Incomplete dominance.
Co-dominance.
Who
Co-dominance.
said incomplete? Who said incomplete? Did I ever say that blood typing is an example of incomplete? No, I don't think I said that, okay? It's co-dominance. Co-dominance means, let me explain the difference between co-dominance and incomplete dominance. Co-dominance means both dominant alleles appear in the phenotype. Like the blood type A, B. A is dominant and B is dominant. And that person has both blood types in his blood. So this is co-dominant, okay? Like a cow, this is my cow here. She has white color and black color. She is blotted black and white, black and white. So this is co-dominance or incomplete dominance, my dear? Huh?
Co-dominance.
Co-dominance. Whoever said incomplete dominance, please understand the difference. Now, incomplete dominance is also a heterozygous genotype. For example, R capital, R small, or it also can be 2 different alleles, or let's use this one so we don't get confused. If I have a red flower that has been crossed with a white flower, the red is considered dominant, so it is given a capital R, and the white is considered recessive, so it is given a small r, but the babies or all the offspring are not red and they're not white. They are pink. They are orange. Excuse me, did someone say something? They are a third phenotype altogether. This is incomplete dominance because they specifically ask you for the difference. You need to know the difference. This is incomplete dominance. So an incomplete dominant or incomplete dominance, this is when you have 2 alleles, one dominant and one recessive, but the phenotype of the offspring is a third phenotype altogether. It is something in between. It's an intermediate phenotype. I hope whoever did this mistake understands it now. It's okay to do mistakes, but it's not okay to repeat the same mistake. Now let's go to the bar graph. Now the example that was given, a bar graph is usually easy because the independent variable is usually not a number, like the blood types A, B, AB, O. And then the frequency of the students with these blood types as my results or my dependent variable. But sometimes they try to confuse you. Let's say, for example, they are asking you to plot a bar graph where the independent variable, I saw this in some of the questions, and that was confusing to a lot of people. The independent variable was the different time during the day. Let's say, for example, at 2 p.m., at 4 p.m., at 8 p.m., at different times during the day, they are counting the number of stomata in the leaf that is or that are open. You know what are stomata?
Yes.
Stomata, these are the openings at the base of the leaf that allows the entrance and the exit of gases for gas exchange. So they are counting the number of open stomata at different times of the day. So the number of stomata that are open, these will be my dependent variable or my results, okay? At 2 p.m., they were 10. At 4 p.m., they were 7. At 8 p.m., they were 4. Now they ask you to plot a bar graph Okay, you will tell me, doctor, they said that the independent variable are times of the day and times of the day are number. So I will make a line graph. No, you are not acting smart here. You are not on the right path. As long as they tell you bar graph, then bar graph. The independent variable is a number, it's times of the day. Again, each one of them will be represented by a bar, a bar, a bar. So we have on the x-axis the independent variable, times of the day. Sometimes it's in the form of a 12-hour clock or a 24-hour clock. And the dependent variable, my results, are on the y-axis. Again, I will divide my graph into equal bars, fine. One bar for 2 p.m., another bar for 4 p.m., a third bar for 8 p.m. Again, the bars should be the same size and equally distant. Okay? They try to confuse you. When they tell you bar graph, it's a bar graph. Every time is a separate bar. Am I clear? Now, some students sent me and they told me that they finished all paper 6 and they want to practice more paper 6. You can go to the lesson plans and do the paper 6 questions over there, because paper 6, they are all the same concept for those who finished all paper 6. Okay, Farah, yes, my dear, you have a question?
Yeah, miss, about paper 4. I'm done with paper 4, but I do want to practice more. So what do I do?
Go to the past papers.
Past papers.
Yes,
Okay.
of course. If you don't have the past papers, you find them all online, but I highly recommend that you have them as a hard copy. And solve the exams that are the most recent. I sent you the 2023. So start from 2023 and move backwards. 2023,
Okay.
2022, 2021. Okay. And when you finish those, I'll tell you what to do. Okay.
Okay.
Now the last graph is the histogram. Now the histogram is actually the easiest and the least common. The histogram, you always have a range and you have the frequency. Okay. And it's very easy because you don't play around with the values. The range will be, let's say, from 70 to 84. And then from 84 to 91. And then from 92 to, or 91 to 97. And so on. So again, the same steps. I'm going to plot my axes, the independent variable and the units. Please don't forget them. And then my frequency on my y-axis. And the range. I can start from 70, by the way. I can leave a small gap and make an arrow that represents the values before 70. I will divide my graph into boxes of 5, and then 70, 75, 80, equal values, okay? 85, and so on. And then my first value from 70 to 84. Okay, so 70 to 84, it will go here at 85. And then from 85 to 91, and so on. And the bars will be fused and they will not be the same width because it depends on the range. Clear? So I hope the graphs are clear. Now, Farah, is your hand raised because you forgot it raised, or you have a question?
No, I don't have a question.
Okay, you have a question? What is it?
No, yes, I don't.
Okay, my dear. Now, investigations. Investigations. Usually, you will be given an investigation in the question. For example, we are studying the rate of germination. Okay, so in the investigation, what did they do? They got 5 Petri dishes and they put 4 seeds in every Petri dish. Okay. And then they changed, this is not 4, and then they got 3 Petri dishes and they put 4 seeds, 1, 2, 3, 4 in every Petri dish, the same type of seed. And they left this or these Petri dishes in the same conditions, in the same environmental conditions. They only changed the temperature by putting it or putting the Petri dishes in different ovens, let's say 20 degrees, 30 degrees, and 60 degrees, okay? And then after 1 week, they calculated or they counted the number of seeds that have germinated. This is what they did in their investigation. Now, what will they do? They will tell you, repeat the same investigation, but don't change the temperature, change the pH. What will you do? Hello? What will you do?
Use the bar.
Excuse me? Hello?
Use different pHs.
Yes, I will use different pH. First, before that, do I invent a different investigation or I use the same investigation and the same steps that they gave me?
Same investigation.
Exactly. I'm not going to invent a new investigation out of my own imagination. I will not do that. I'm going to go to the same steps and I will copy and paste the same steps. They said they got 3 petri dishes. Okay, then I will get 3 petri dishes. They added 4 seeds. I will put 4 seeds in every petri dish. They will expose the petri dishes to the same environmental conditions. Fine. What did they change in the investigation? The temperature. Now that will be kept what? Constant. This is the only thing I will change. Instead, what will I change? The pH, because this is what they asked me to do. They want to know, how will you change the pH? Now, how do we change the pH?
pH buffer.
Exactly. Either if you don't know what is a pH buffer, it's fine. You can say I'm going to use a strong acid or a weak acid, a strong base or a weak base, and try to give examples. Or if you don't want to say that and you want to be very professional, I'm going to use a pH buffer solution. Some of you still don't know what this is up till now. This is a ready-made solution in the lab that has a fixed pH and its pH does not change throughout the investigation. So I'm going to use pH buffer solutions. One of them will be a weak acid and a strong acid, weak base and strong base. Now, I think you know, give me a value for a weak acid.
3 or 4.
No, 3 is a strong acid. The pH values are from 7
12.
to 4. Yes, 5 is a weak acid. 6 is a weak acid. Strong acid is from 1 to 3. Okay? A strong base is close to 14. And a weak base is close to 7. Like a weak base is, let's say, with a pH of 12. No. pH of 8, 9. Okay? So give examples. I will change the pH and all the other steps will be the same. Now, in order to score the 6 marks, please write the answer in an organized manner. My variables first. What are the 2 variables that I have to mention?
Temperature.
My variable,
Independent.
yes, my independent variable, 2 dots. I will write my pH. I will change the pH. My dependent variable, 2 dots. I will get, or I will count the number of seeds that germinated, okay, after a week. I did not make it up. I will go get it from the investigation and write the full word, please. And then my constant factors are very important. What will I keep the same? So I have to write the word, I will use the same seeds, the same temperature, excellent, the same time of exposure, the same type of seed, number of seeds, the same everything, okay? Same time of day, same sunlight exposure, whatever. And then I will write my steps. Now, this is the easiest part because I will simply copy paste it from the investigation that is already given. I will not make it up. I will do the same steps. I will get 3 petri dishes with 4 seeds in every petri dish. I'll put them in the sun. I will change the pH. That's the only thing I will change by adding different buffer solutions. And then I'm going to count after a week how many seeds germinated. Now, if you want to add a control, go ahead. If you don't want to add, you do not lose a mark for no control. But if the control, I'll give you a hint, if the control involves enzymes or any plant or seed, the control will be boiling the enzyme or boiling the plant or the seed. Never say that the control will be using room temperature. And I can also, in an acid condition, acidic conditions, I can use the control here, neutral pH. If you're not sure of the control, you can avoid it. You don't lose a mark if you don't write the control. Okay? Now, the last 2 sentences that are the final sentences, what do I say at the last or the end?
The safety precautions.
Exactly. I will repeat and calculate average, and I will wear gloves for safety, and I will be done. Okay? 1, 2, 3, 4, 5, and 6. These are where the 6 marks go. I hope it's clear. Now, again, classic questions are, what is an error? And yes, sometimes they will ask you to create an investigation that they did not give you before. An investigation like this will always be related to a food test. carry out an investigation to test for the presence of protein, for example, at different temperatures. And this will be not preceded by a previous investigation. Why will they do that? Because you are expected to know the different food tests. What is the food test for vitamin C? Yes, Farah. Yes, the vitamin C, why am I revising it? Because again, the vitamin C, the DC pip has to be in the test tube. The DC pip is blue in color. And then you keep on adding drops of the vitamin C, drop by drop, a drop and you shake, a drop and you shake. And suddenly this blue color will change to what?
Colorless.
Colorless. Okay? And you count the number of drops. The more the drops, the less concentrated the vitamin C and vice versa. And you will change the independent variable depending on what they tell you. If I will change the temperature, I will use different thermostatically controlled water baths and give examples. And never use a room temperature water bath as a control. For a control, what will you do if you want to write a control? Yes, Ahmed.
No vitamin C.
Yes, no
I have
vitamin
a
C,
question,
water.
miss.
Yes, my dear. Ask whatever you want.
When do we use rate and when do we use the number of drops as the dependent variable?
It depends on the question. If the question is giving you a previous investigation and they're asking you, or they're in the investigation, they're calculating, or the results are a rate, then use the rate. So you just follow the investigation. The drops here, in this type of question, there is no previous investigation. Here you should know the food test. If they ask you for the vitamin C test, here we count the number of drops. Okay?
Okay.
Fine. Now I want to revise also an experiment that a lot of you get confused, I don't know why, which is the effect of the amylase enzyme on the breaking down of starch. Okay? I will put both the enzyme and the starch in the test tube. So in this test tube, I have amylase and starch. And then what do? Who will remind me? Do I add my iodine directly to the mixture?
No, you use a spotting.
Exactly.
Take
The
drops at
iodine-
time intervals.
Perfect. The iodine, I cannot add the iodine directly to the mixture because I will ruin my mixture. So what do? I will get a spotting tile, something like a tray with small depressions. And in every depression, in every hole, I already have iodine. What is the original color of iodine?
Orange, red.
Red brown.
Yes. No, it's not red. It's orange brown. So they are all orange. Every minute, I'm going to get out one drop from my mixture and put it on my drop of iodine on the spotting tile. And I will look at the color change. If it changes to blue-black, this means starch has been broken down or not yet?
Not yet.
Not yet.
Broken
So
down.
after another, no, Habibi, it did not break down.
Oh, صح.
Okay, my dear, don't get confused. And then another minute, I'll get another drop. And then another minute, and then get another drop. And I will keep on doing that until the color of the iodine remains what?
Orange.
Very good, remains orange. Here I will know how long did it take for the starch to break down. As long as time involved, this is a rate for whoever was asking. Okay? Now, how can you improve an error? Again, these are very easy questions. Any reaction or any
Colorimeter.
idea? What did you say?
Colorimeter.
Yes, correct. Any investigation or experiment, and all the food tests actually, that involves color is an error. If I use a dropper, it's an error. If I am counting the number of bubbles, it's an error. All these are inaccurate. For color, sometimes I can see color differently, so I'll use a colorimeter, correct. Colorimeter. A dropper is inaccurate. I'll use something that is graduated, like a measuring cylinder, a pipette, a burette, anything with a ruler. Okay? Bubbles, no. I will use
Volume.
a graduated syringe. What did you say?
I said you measure the volume.
Correct. Perfect. I will measure the volume with a syringe. Anything that is graduated, because bubbles Of course, you can miscount and the bubbles are not the same size, etc., etc., etc. And I want to just go over the term anomalous. What is an anomalous value?
A value
Altogether
that
different.
doesn't match the rest.
Very good. If you have values 1, 2, 1, 3, 5, 1, 2, 3, what is the anomalous? What's the anomalous value?
12.
It's the
12.
5, of course. The 5 is very different. Okay. Okay. Very good. Now, again, let's revise very easy calculations. What is, or how do I calculate average?
The values over the number.
Yes, the total over the sum. Percentage. Percentage is very similar to the average, but I add another step.
Times 100.
Yes, exactly. Total
Fine.
over the sum, I multiply by 100. Don't get confused. Everything is different. How do I calculate the percentage change?
Final minus
Um,
Just
initial
initial over
initial.
initial times 100.
Exactly. Okay, you are saying it and you're very confident and sure of yourself, and then you do mistakes. You know why? Because you do not determine what is the final value and what is the starting value before you start. Okay, so you write the formula, you always give me the correct formula, but a wrong answer, and you lose one of the marks because you do not determine what is the final value and what is the starting value. And please read the question. Okay, you will know from the question which is the final and which is the starting value. Very good. Now also please read the question, because if they tell you give an answer to 2 decimal places is different. Yes, Hadia.
Doctor, when do you use heat and in investigations?
In what, my dear?
In investigations, when do we use heat?
Someone tell me, someone answer her question. Which is the only food test in which we use a water bath?
Benedict.
The Benedict. The Benedict and the reducing sugars and the glucose. Okay, my dear?
Okay.
And it should be nearly, or it should be at least 80 or 90 degrees. Now, the 2 decimal places, you give a different answer than when they ask you to give an answer to the nearest whole number. Whole number means no decimals. 2 decimal places means 2 decimal places. For example, if you got the answer 3.6666, how can you give me an answer to 2 decimal places?
3.67.
Yes, correct. 3.67. Why did we round to 7? Because the adjacent decimal here is more than 5. 5 and above, you round up. 2, okay. 2 whole numbers or a whole number?
4.
4. Correct. 3.6. Okay, then this will be 4. So please read the question. Just read the question to avoid losing a mark here and a mark there, etc., etc., etc. Okay, I think I am done with paper 6. Let me do some general revision. Yes, what do I want to talk about? Yes, potatoes. Potatoes are very important, movement in and out of cells. If they give you, because some of you are not very okay in math, if they give you a potato that they put in different concentrations of sugar or salt, one concentration is 1.0 mole per decimeter cubed, and the unit is not our problem now. And then another concentration is 0.5 mole per decimeter cubed. And then a third concentration is 0.1 mole per decimeter cubed. And then a fourth concentration is distilled water. Now, to my surprise, some students don't know what distilled water mean. What does distilled water mean?
Pure water.
Yes, yes, distilled water is only water or pure water, pure water. I was very surprised when I realized that some people don't know what distilled water. They try to play around with the words. Distilled water is the same as pure water, okay? Now, from those 3, A, B, and C, which concentration is the highest? It has the most sugar. A or B or C?
A.
Yes, in A. And which concentration is the least concentrated?
C.
Distilled.
Distilled, yes, you are correct. I'm choosing between A, B, and C. From those 3, which one has the least? Yes, C. Okay? So 1.0 is a number value more than 0.1. Fine. Now tell me, when this potato is put in solution A, which is very concentrated, what will happen to this potato, or what will happen to the plant cell?
Will shrink.
Shrink.
Yes, the vacuole will shrink. Be specific. The vacuole will shrink. And what will detach away from the cell wall?
Cell membrane.
The cell membrane. And what are the keywords that you write here?
Plasmolized.
Very good. Flaccid, plasmolized. Now the potato itself, will it be hard, or the
Soft.
potato will be soft and floppy? Yes. When a plant or a potato loses water, it will be soft or floppy. Sometimes they use the word floppy. Okay. So it was going to be soft. In which solution or in which concentration, A, B, or C, forget the distilled water now, will the potato be hard and increase in mass because it will absorb
C.
water? B or C?
C.
Why?
C.
C is the least concentration. C is nearly pure water. Now, see, this is what I'm saying. You don't know that 0.5 is a bigger value than 0.1. 0.5 means I have more sugar. 0.1 means I have less sugar and more water. Okay, my dear? Who got confused? So please look at the numbers. This surprises me. You have some values like, again, I will use the same examples, 1.0, 0.2, 0.4. Which is the most concentrated? 0.1% or mole per decimeter, or 0.2 or 0.4? A, B, or C?
0.4.
No, is 0.4. 0.4 is a bigger value, or 1 is a bigger value?
1.
1. So the
Sure.
1 is the most, yes. See, it's okay. Everyone gets confused. That's why I'm revising that part. So the problem is, if you get confused, the whole answer will be incorrect. So please read the question carefully. You usually get confused because you don't read carefully. Be patient and read the question carefully. So a value of 1.0 is the most concentrated in those three examples. And a value of 0.2 is the least concentrated. Okay? 0.4 is in between 1.0 and 0.2. Okay, look, if we have a line graph, you have 0, 0.1, 0.2, 0.3, 0.4. So the 0.4 here is more concentrated than 0.2. 0.2 is closer to the 0. Okay, I don't know how to teach math. This is math. And 1 has a higher value than 0.2 and 0.4. I can't believe that I'm explaining that. Okay, any questions? And when a potato is soft, sometimes they call it floppy. Now, I think I revised what I wanted to revise in paper 6.
I'm gonna say one. I have a question.
Ask, my dear.
If يعني لو البوتيتو is put in a 0.4 concentration هتبقى flaccid ولا turgid?
It depends. You are comparing it with what? They will always give you other concentrations. If you have several examples, 1.0, 0.2, and 0.4. If you put it in a concentration of 1.0, it will be very flaccid because it will lose water, because this is the highest concentration. 0.4, it will also be flaccid, but not as 1.0. And 0.2,
Will be turgid.
Yeah, it will be turgid. It depends. They will have to tell you there's a change in mass, there's a decrease in mass. They will have to give you a hint. Okay, they will not leave you.
Thank you.
You're welcome. Okay, now, what do you want me to revise?
Doctor, can you give us general revision about inheritance?
About what?
Inheritance.
Inheritance? Okay.
Yes.
Now, when I talk about inheritance, I'm going to talk about two things. The sex-linked. Okay, I think that this is the hardest part. And the test cross. I think the rest are easy. Cross. Okay. Now, the sex-linked. These are diseases or alleles that are found where? On which chromosomes?
Male chromosomes.
What did you say?
Male chromosomes.
No, you're wrong.
X chromosome.
On the X or the Y? X or the Y? On the sex chromosomes. Your answer should be sex-linked diseases or alleles are found on the sex chromosomes. I have two sex chromosomes. I have the X which is long, and I have the what? The Y,
The
which
Y.
is very short. The X is the female chromosome, and the Y is the male chromosome. Usually, those sex-linked alleles, they will be found where? On the X or the Y?
On the X, I think.
On the X, yes. Why on the X? Because it is longer or shorter?
Longer.
What do you see? Yes, it's longer. And because it's longer, it has more space to carry additional alleles. Usually those alleles are for diseases. Now, you should know two diseases or one that usually comes in your exams. The color blindness. Color blindness, this is a sex-linked disease, and it is recessive. Okay? This is always found on the X chromosome. And because of that, it is always more common in males. Now let's revise why it is usually common in males. Who will tell me the genotype for a colorblind male? A male that is colorblind.
X, B small and Y.
Yes, XY male. And he is colorblind, so I will put a small letter on the X. Any small letter that represents that he is colorblind. Now, this male, he will be sick or healthy?
Sick.
He will be sick, even though he has only one recessive allele. Males cannot be carriers in sex-linked diseases. Males are either sick or healthy. It's only the female that can be a carrier. Can you tell me the genotype of a healthy carrier female?
X, B capital and B small.
Excellent. B capital, B small. Okay? Now, can you tell me the genotype of a sick female? She is,
X, B
Yes.
small and X, B small.
yes, and this genotype is actually rare. That's why it's not common for sex-linked diseases to have to be present in females, because they have to inherit two recessive alleles, and this is very rare. She's very unlucky if she will inherit two recessive alleles. In males, only one recessive allele is enough to cause the disease. Okay, I have different colors and I'm exploiting the situation. Okay. Now the test cross. Who will remind me, what is the test cross?
When there's one unknown genotype, you
Right.
link it with a recessive genotype.
Yes, now this unknown genotype, what phenotype is he showing me? Dominant or recessive?
Dominant.
Of course, because a recessive phenotype has one genotype, which is homozygous recessive. This unknown parent is a dominant parent. He's showing me the dominant phenotype, like he's tall, but I don't know if he is homozygous dominant or he is what? What's the other option? He can be heterozygous. Now, what do? We will do a test cross and we will check all the what? We will look at I wrote it on the board.
The offspring.
The offspring. If all the offspring, if 100% of the offspring are tall, then that parent was homozygous dominant or heterozygous?
Homozygous dominant.
Both.
Homozygous dominant. Who said both? Who said both? Who said both? Homozygous dominant. Why homozygous dominant? Because all the children will then have one dominant allele. Even if they will inherit a recessive allele from the mother, they will have one dominant allele from the father. So all the children, if they have 100 children, all the children will be tall. But let's say 50% were tall and 50% were short. So what was the genotype of this unknown parent?
Heterozygous recessive.
He will be
Dominant.
heterozygous recessive. What's heterozygous? He'll be heterozygous. Just it's not called heterozygous recessive, it's called heterozygous. So if the offspring 50% were tall and 50% were short, then that parent was definitely heterozygous. If you are confused, the only thing that will help you is to create a Punnett square. I have a heterozygous parent crossed with a homozygous recessive parent. I will, excuse me, what happened here? I will create a Punnett square. I'm going to divide this gene into its two alleles, one dominant T, one recessive T, and this parent. I'm going to make one small t, another small t. Please don't write anything here. Okay? This child will be heterozygous. This child will be what? Homozygous recessive. I hope you're with me. This child here will also be heterozygous, and the last child will also be homozygous recessive. How many out of the 4 are tall?
50%.
Yes, those are tall. And how many are short?
50%.
Again, 50%. So I have 50 tall, 50 short. As long as I have short children, this means that this parent that was tall, he had recessive alleles in his genotype and he was heterozygous. Okay? Now let me revise. I want to revise something because I don't know why a lot of you are not getting it. The difference between natural selection and artificial selection. Can someone remind me what is artificial selection first and what are the steps?
When people choose the parents and breed them.
Correct. So if you will break down in steps, please study the steps because this question is usually out of 3 or 4 marks. Go to variation and selection. Now, artificial selection, yes. First, you start with the word, the farmer or the scientist or the people they choose. They are the ones
Parents
who
with the
they
desire.
choose. Yes, they choose the parents with the desirable characteristics or the favorable characteristics. And these parents will, yes, continue, my dear.
They will breed them together,
Yes.
and then they will choose offspring with desired characteristics and breed them together also.
Very good. That's it. It is so simple. You just have to mention that the practice will be repeated and repeated and repeated. I keep on repeating it until all the offspring I have on the farm have inherited the favorable characteristics. I want to ask you a question. In artificial selection, was there any type of competition? No competition. No competition happens here. The competition, where does it happen? In the artificial or the natural selection?
Natural selection.
Natural selection.
Correct. Can someone remind me, what are the steps of natural selection?
It starts with mutation.
Very good. It starts with variation. And this variation is the result of what, my dear?
Mutation.
Yes, correct. Okay. And then what will you say?
When organisms reproduce more offspring than the environment is capable of, I don't know.
Supporting them. Yes, I have so many offspring. So when I have lots of offspring and limited food or limited space, this will create what?
Competition.
Excellent. Do they all survive?
No, organisms with the adaptive features survive and reproduce. And
thank you so much. Those with favorable features, it's actually very logic. Those with favorable features will survive and they will reach reproductive age and they will breed and their favorable genes will be passed on. Okay, so these genes, mentioned that the word genes, will be passed on so that all the population will, with time, will inherit the favorable characteristics, and this will create natural selection. And when the organisms change with time, this is called what?
Evolution.
Correct, evolution. Okay, so natural selection is a longer process. Which is slower, the natural or the artificial?
Natural.
Yes, which will give me more variation?
Artificial.
Natural. Yes, the artificial actually, it is faster. Yes, this is an advantage, but it affects the variation. It reduces variation or biodiversity. Okay, what are the disadvantages of sexual reproduction? We always revise the advantages of sexual and the advantages of variation. Don't forget that sexual reproduction also has disadvantages. What are they?
Needs gametes, so lots of energy.
Yes, I need more time and I need more energy.
Requires a mate.
Yes, I need a partner. I cannot isolate in the wild. Okay.
Fewer offspring than asexual.
Yes, I have less offspring, correct. And there is a higher chance that the unfavorable characteristics can be passed on. If I have asexual reproduction and I have favorable characteristics, then these favorable characteristics will definitely be passed on. But in sexual reproduction, I always give you the example of a very pretty mother, okay, who will have a daughter that is as pretty as she is. This is the disadvantage because some unfavorable characteristics has been passed on. If that mother produced by mitosis, her daughter will be a replica and will be as much as pretty as the mother. Okay? Now, don't forget osmosis and its definition. It's a 3-mark definition. It's the net movement of what is osmosis. Why is it out of 3 marks?
It's the direct movement of from high water potential to low water potential,
Across
right? Across the
Semi-
concentration gradient.
No, across a semi-permeable membrane. So why osmosis is out of 3 marks? Because I am talking only about water in the liquid state, and it moves down a concentration gradient from high to low and across a semi-permeable membrane. Okay, now also something I want to revise because it confuses a lot of you. The hormones in the menstrual cycle. I revised it in class, but I will revise it again. Again, the menstrual cycle, 28 days. Fertile period is around the ovulation, day 14. And I tried to help you remember the first 2 weeks, I am producing and releasing the egg. So from day 1 to day 14, I am producing a new egg and releasing it. From day 14 to day 28, I'm waiting for the sperm. The sperm comes, welcome. The sperm does not come, it's okay, I will survive. Fine, I will go out of the body. So from day 1 to 14, the egg is being produced, a new egg starts to develop, and then it is released. And then from day 14 to day 28, what is happening? I'm waiting. I don't wait for the 2 weeks. The egg only waits for 2-3 days only. The sperm comes, fertilization will happen, and mitosis and development, etc., etc. The sperm does not come, what will happen? The egg will move down the fallopian tube, the unfertilized egg. It will go to the uterus, and then it will come out in the form of blood. Now, anyone has any questions? I will revise the hormones. Hormones. Believe me, they're very easy. The follicle stimulating hormone, what does it do?
Releases the
Growth and
egg.
development of follicle.
Help in the development of the follicle.
Matures the egg.
You are all correct. One of you said matures the egg. One of you said helps in the development of the follicle. Yes. In case you forgot, read the hormone from back to right. It's a hormone that stimulates the follicle. I hope you know what the follicle is. These are the group of cells around the egg that will help in its growth and maturation. Okay, I have another hormone, the luteinizing hormone. It has one function only. What is it?
Ovulation.
Releases ova to the oviduct.
What did you say? Oh, it released the egg in the oviduct? Yes, which is ovulation, correct. And I want everyone to join, please. Now we have another hormone, which is the estrogen.
It's built up in the endometrium.
Yes. The estrogen, in normal situations, it is responsible for the secondary sexual characteristics. And in the menstrual cycle, the estrogen is the pre-ovulation hormone or the post-ovulation hormone?
Pre-ovulation.
Yes. And it also maintains the thickness of the endometrium or now the progesterone, it is the post-ovulation hormone or the pre?
Post-ovulation.
And what does it do? It has one
Maintains
Maintains
thickness.
thickness of the endometrium.
same function as the estrogen, correct. Maintains the thickness. Okay, let me see. Don't forget the foods and the importance of vitamin D and calcium and iron. It's so easy. Okay. Who has any questions? Let me look.
Miss,
Yes, my dear.
Miss, I still don't understand the LH and the progesterone.
Okay, my love, I'll tell you. LH is a hormone that releases the egg from the follicle. This is my dear, the female system. This is my female system. She looks like a clown. Okay? This is the clown. Doesn't it look like a clown? This is my ovary. And the ovary has many eggs. Only one egg is produced per month. This egg will grow and grow and grow during the first 14 days of the menstrual cycle. And then after it is ready, it needs to be released. The release of the egg into the oviduct is called ovulation. But it needs permission from the brain in order for it to be released. The brain will release a hormone that will allow the release of the egg. What is this hormone?
Luteinizing hormone.
Yes, my dear. So the luteinizing hormone is released from the pituitary gland in the brain, This luteinizing hormone is responsible for ovulation. Only, this is its only function. Now, the progesterone hormone has another function. Every hormone has one function. The progesterone, it starts with the letter P. It is the hormone that keeps on increasing during pregnancy. During pregnancy, the walls of the uterus, they have to remain very thick because this is the baby's house. The baby is sinking in the walls of the uterus, and it is receiving from the placenta and the umbilical cord the food and the oxygen and the glucose and the calcium and the minerals, etc., etc., etc. So if the uterus breaks down, it will kill the baby. So the progesterone has a very important function. It keeps the walls of the uterus nice and thick or thin?
Thick.
Thick. This is the function of the progesterone. The other two hormones, follicle stimulating hormone, from the name, it's a hormone that helps the growth of the follicle, which is the same as the egg. It is the hormone that helps in the growth of the egg. Anything easier than that? We have the last hormone, the estrogen. The estrogen, think of the estrogen hormone as the hormone that makes a female look like a female. The more estrogen, the prettier you are, the more you will be of a female. It's a hormone that is present all the time because it's responsible for your secondary sexual characteristics. Okay? It makes you a female. I think I'm clear. Also, the estrogen increases during pregnancy because it does the same thing the progesterone does, but before ovulation. It keeps the uterus thick or thin?
Thick.
Thick. Correct. The follicle stimulating hormone and the luteinizing hormone are produced from the pituitary gland in the brain, and the other two hormones, estrogen and progesterone, are produced from the ovary. And the progesterone is produced from the placenta in the late stages of pregnancy. Did I make things easier?
Yes,
Miss, I have a
doctor.
question.
Ask.
I don't know what is the corpus luteum.
Okay, the corpus luteum, who will tell her?
The yellow body.
Yes, in the ovary, I have eggs. Eggs are surrounded by follicular cells. These follicular cells are responsible for the egg. It's like they are guarding the egg. They are responsible for the growth and the development of the egg. The egg keeps on growing and growing and growing until at a certain point, this egg is released into the oviduct. This is
Okay.
oviduct. What remains in the ovary? Those follicular cells. These follicular cells, after the egg is released, these follicular cells change into another structure known as the yellow body or the corpus luteum. And the yellow body is the structure that produces the progesterone hormone. Better?
Okay, yeah, thank you.
You're very welcome. Let me look at the multiple choice questions. After a reaction, what remains at the end of an enzyme-controlled reaction? We have enzyme that reacts with a substrate to give me products. Now, does the enzyme go away?
No.
No. I have
Unchanged.
reacted, sorry. Yes, the enzyme doesn't go away. The enzyme remains and the product remains. What goes away? The substrate, because the substrate has been used up. Now, what makes an enzyme specific?
The active site.
Yes, the active site, not the temperature, not the pH, the active site. And don't forget the graphs for apparent temperature and pH. Okay, okay, okay. What are the complementary base pairing?
A with T and G with C.
Yes. Now I see that biology is the nicest lesson and it's the easiest lesson and it is the best lesson or the best subject. Anyone has any questions?
Doctor,
Yes, my dear.
can you revise the HIVs?
Okay, very, very easy. Who will remind me? I forgot. HIV, what is it?
Dormant stage of AIDS.
Okay, can you please give me a second until I open this door? Just 1 second. I'm not going anywhere. Don't go anywhere. Okay, HIV, my dear, It is a sexually transmitted disease. In other words, it's a disease that is transmitted via intercourse. So we need to protect ourselves from these diseases. We have so many diseases that are transmitted via intercourse, but HIV is the most common one. It's the most dangerous because there is no treatment for HIV. HIV is caused by a virus, not a bacteria. So can we treat it with antibiotics?
No.
No, okay? There is no treatment actually for HIV. HIV is caused by a virus, and when you are infected with HIV, God forbid, it does not kill you straight away. It gives you, or it takes years. Fine? The HIV, when it infects you, you will be HIV positive. Until it actually causes disease, you will be, or you will have AIDS. Again, 1 second. Okay, I'm back. So when you are infected with HIV, God forbid, you will not be sick immediately. You will be HIV positive. In other words, the virus is in the body, but the virus is sleeping. The virus is inactive. This is said to be dormant. And then at a certain point, when your immune system becomes very weak and you start showing signs and symptoms of the disease, this is when when the virus becomes active, and now you are said to be an AIDS patient. Now, HIV is transmitted through intercourse, transmitted through blood or blood-borne products. So what do you do in order to protect yourself from such a disease?
Not sharing needles.
Don't share needles. Don't share anything that comes in contact with blood, not only needles, like nail clippers, like shaving machines, like toothbrushes. You have to have protected sex by using condoms. Okay, or avoiding random intercourse altogether. All these can be written as answers. HIV can be transmitted also from the mother to the baby during pregnancy, through the placenta, while she is giving birth, during breastfeeding. Okay, so these are what you should know about HIV, and we will go back and study the HIV and the diseases in a lesson called immunity. Who wants me to revise anything else? I think we revised the digestive system in detail in class. Anyone has any questions?
I just want to make sure if like the sentence اللي هقولها صح ولا غلط.
Tell me, my
The placenta from 4th month till delivery releases the progesterone, صح?
The placenta, what, my dear?
From 4th month till delivery releases الهرمون progesterone.
Yes, what you're saying is true. Yeah, the end of the 3rd
Okay.
week, the starting of the 4th month, correct. Just
Thank you.
late stages of pregnancy is enough. Anything else?
Doctor,
Yes, my dear.
can you revise the protein synthesis?
Yes, I can revise, of course, the protein. I have a nice cell here. I have a nucleus, and I have DNA. And I want to copy part of this DNA because I want to make one protein, not all the proteins. Who will copy? How will I copy it?
Messenger RNA.
Yes, so part of the DNA is a large molecule, by the way, and this is coiled up, it's very big. It has many, many, many genes. I want only one gene. So I'm going to copy that gene on a messenger RNA molecule. So that's the first step. Step number 1 is called the transcription. Transcription is copying part of the DNA on a messenger RNA molecule. The DNA remains inside the nucleus. The DNA does not go out because it's big. Only the messenger RNA will go out. It will go out through an opening in the nucleus called the, remind me.
Nuclear port.
Very good, the nuclear port. This is step number 1, finished. And then the messenger RNA will go attach to another organelle that will make proteins. What's the name of this organelle?
Ribosome.
Yes, ribosome. So this will be step number 2. It will be called translation. You have to mention in that step that the messenger RNA will attach to the ribosome. And the ribosome, what will it do?
Translate the code.
Very good. It will translate the message. And don't stop here. It will translate the message. In other words, it will assemble What does it assemble? The ribosome assemble the what next to each other?
Amino acids.
Very good.
Amino
Every
acids.
codon, no, I don't want to go to the details. Every 3 bases on the messenger RNA attracts a specific amino acid. These amino acids will then join together to form a chain of amino acids or a polypeptide, okay, which is my protein. And the sequence of amino acids is determined by the sequence
The
of
base
bases
sequence
on
of the
the
amino.
excellent. Okay, so these are 2 steps: transcription, translation. Transcription, I will make a copy of the DNA on the messenger RNA. Translation, I will read it and I will create a sequence of amino acids, which is my polypeptide chain. Who has any
Doctor?
questions?
Doctor,
Yes, my dear.
I don't understand what alleles are.
Okay, I'll tell you what alleles are. You have how many chromosomes in your cell?
46.
46. These are 2 pairs. I have 1 pair or 1 set of pairs. In other words, I have 23 maternal. Maternal means from the mother or the father?
Father.
No,
From the mother.
maternal starts
Mother.
with M, comes from the mother. And 23 paternal. Paternal means mother or father?
Father.
Father. Good. Now I have, let's say, I will look at just 1 pair. This is 1 chromosome from the mother and another chromosome from the father. These chromosomes have lots of genes. Every gene codes for 1 characteristic or 1 protein. If I look closely at this gene is made up of how many parts? 1 or 2 parts? What do you think?
1.
Why? This is 1 chromosome and this
Oh,
is
2.
yes, okay. The chromosome is divided into genes. If I look closely at every gene, this gene is made of, let me change color. This gene is made up of 2 smaller parts. 1 small part is called the allele. So this is an allele. You inherit 1 allele from every parent. What is the definition of an allele? These are the different examples of a gene. In biology, we say it this way, the alternative forms of a gene. In other words, this allele can be dominant. It can be recessive. If you have a gene for height, you inherit 1 dominant allele from the father and another dominant allele from the mother. So now you have a gene of homozygous dominant alleles. You will be tall or short?
Tall.
Tall. So what is an allele, my dear? The allele is what do you inherit from every parent. Together, 2 alleles make 1 gene. A gene is made up of 2 alleles. 1 allele from the mother and the other allele from the father. Is this better?
Yes, thank you.
You're very welcome. Who has any other questions?
Okay.
Yes.
Can you explain the different types of dominance?
Yeah, I will. I said it, by the way. We have 2 types of dominance. We have co-dominance and we have incomplete dominance. That's it. We have 3 types of a heterozygous condition, but 2 types of dominance. 2 types of the co-dominance means both alleles appear or a 3rd phenotype appears.
Both.
Both. Co means 2, both. And the easiest and the best example is blood type A, B. A is a dominant blood type and B is also a dominant blood type. And only in the case of co-dominance, we will use 2 different capital letters. I am blood type A and B. So I have both dominant alleles and they both appear in my blood type. So I am co-dominant. Nice? Now, incomplete dominance. I'll give you a nice practical example. I have a tall father and I have a very short mother. By the way, this is not fair. No, I have a tall mother and a short father. A tall mother and a short father. And you, this is you. What's your name?
Stuff.
Mustafa. Okay, Mustafa, you are not tall and you are not short. You are average. So this is co-dominance or in between?
In between.
Yes, this is incomplete dominance. So incomplete dominance, my dear, this is when you're not dominant and you're not recessive. You're something in between. Okay?
Okay, thank you.
You're very welcome. Who wants me to revise anything? Okay, if you have any questions or you're not sure of anything, you can send me on WhatsApp. And when I see the message, I'll do my best to try to explain it. Fine? We revised all the lessons, I guess, for year 10. Do not ignore year 9. Yes, there is a question that a lot of you did or you got wrong in the last quiz, which was the definition of the haploid cell. Some of you said in your answer, it is a cell that has 23 chromosomes. This would be considered incorrect. Why? Who will tell me why? Do all the cells in all human beings, all haploid cells have 23 chromosomes? Or this number is only for humans?
This number is only for humans.
For humans.
Exactly. A haploid cell, the definition is a cell that contains one set of unpaired chromosomes. If you said in your answer, it's a cell that contains half the number of chromosomes, yes, I will consider that correct because it's the half. But don't give me a specific number because that specific number is for a particular species only, which are humans. Dogs don't have a total of 46. Elephants don't have a total of 46 and their haploid is 23. Every species, every organism has a different total number, has a different haploid and a different diploid number. Please pay
Half
attention.
of a diploid cell is correct ولا لا?
Excuse me?
Half of a diploid cell, is that correct ولا?
The word half
Diploid
will give you
nucleus.
the answer. If you just mention half, the word half
Okay.
gives you the answer. But don't give a number. And please use the original definition, one set of unpaired chromosomes, because that's the only answer that I saw on the mark scheme. Okay? And what else that most of you got wrong?
Can I mention 23 chromosomes as an example? يعني
As
after
an
I
example, yes. As an
For
example.
example, يعني the haploid is one set of unpaired chromosomes. For example, 23 chromosomes in
Exactly.
humans.
This is perfect that you gave me the definition and an example. This is perfect. Always give examples when you can. Okay. And the implantation also, I don't know why most of you got it incorrectly. When a zygote is implanted, it is not going to be implanted in space. A zygote is not going to be planted. It's going to be implanted in the endometrium. So the answer was the V, the endometrium, not here. Okay. Most of you got it correct, but some of you did not. And then also this common question that drove me crazy. Why should the gametes be haploid? Why is this very important?
So
Imagine,
diploid number is restored after.
yes. Imagine you have gametes that are 46 fused with 46. The zygote, how many chromosomes will it have? It will double. The zygote will have 22 chromosomes. You will create a different species. The haploid, they have to be haploid. They have to have only half the total number of chromosomes because when they are fused at fertilization, the diploid number is restored and it does not double. Okay? These were the common 3 mistakes. Again, I'm going to repeat. If you have any questions while you're studying, do not be embarrassed to ask. Embarrassment will not help you. Ask me. If you want me to explain a whole lesson, I will send you a video for that particular lesson. If you want me to explain something in specific, I will explain it to you. Okay? Don't leave yourself like this. Don't ignore anything. While you're reading the notes, I will not tell you memorize word by word, but understand every single word. Understanding is very important. Okay? Anyone has any questions?
Doctor,
Yes.
can you explain the sources of genetic variation?
Yes, I can. MMRR. The first M is what?
Mutation.
Mutation. What is mutation?
Change in the base sequence.
Yes, of the DNA. I hope it's clear. In other words, a change in your genes. Of course, it will create different organisms. The other M is what? Which type of cell division?
Meiosis.
Meiosis. And don't forget to study the definition of meiosis. It's a 3-mark definition. Reduction division that produces genetically different haploid cells. Now, what is R? Random what?
Mating.
Yes, random mating. Do we know which male will mate with which female?
No.
No. Okay. And then the last R is random what?
Fertilization.
Correct. Do we know which sperm will mate with which egg?
No.
No, this is something that is very spontaneous and very random. And all these things, all these factors causes variation within organisms. Of course, only the organisms that reproduce by sexual reproduction. Okay?
Okay. Thank you.
You're welcome. Anyone has any questions before I go?
Just to make sure, in the continuous variation, it can
Yeah.
be represented as a line graph and an histogram, right?
Correct.
And in the discontinuous, bar graph, right?
Only bar graph, no any other option,
Okay.
because I have limited phenotypes. No intermediates, no categories in between. Correct. Okay. Any questions? Anything that crosses your mind? Ask me.
Yes, can you explain some paper 4 inheritance questions?
Yeah, okay, if you are going through a question and you stop at a question, send me the question and I'll help you.
Okay.
Okay. See you.
بس
Yeah.
هو الانترسكس تكون مكتوب فوق صح؟
Yes.
Okay.
Okay. Study well. It will be very easy if you study well. Enjoy your time.